Microbialization. How does it affect wild reefs? Can it be controlled in aquariums? This really came about as I was trying to get someone who I'll be talking about in the talk, Forrest Rower, to attend and speak at this convention. Unfortunately, I wasn't able to get him here. He wasn't available. I was able to talk with him at length. And then I decided, well, gee, I got invited to come here and I could talk. Oh, my most recent lecture that I gave was about giant clams, and that might be interesting to this group. But I thought, you know what, let me tackle this in a way as best as I can to introduce this topic, because I've known for more than 10 years that this was something that we should be talking about and investigating, but nobody was really talking about it until rather recently. I saw Salem brought up the topic on reef builders, and I thought, well, yeah, now it's starting to catch on. We should bring it up again. Anyway, as you've just heard, Two Little Fishies is my company, and I've seen most of the presenters have the logo and contact information on all their slides. I just have one slide here at the beginning. If you want to reach me, email is the best way. Calling, it's really hard. Thanks, Matt. I wanted to mention Jen Bowen and Cody Sharp, neither of whom I had met previously. I really enjoyed their lectures yesterday. And you might think that they were talking about the same topic as I am. Actually, it's a related topic, but not the same. So I think when you hear microbialization, you're probably thinking that I'm going to be talking about bacteria, which I am, and antibiotics and probiotics and how do we deal with this in our aquariums. And that's not really on target. That's actually off target. So a little bit of backstory. I think it was, let's see, 2015 or 2016, I was speaking with Frank Malero, Dr. Malero at the University of Miami, because I was working on two products. One is the Accuracy Reference Solution, and the other was Accuracy 1 Seawater Mix. And he was helping me out, and he had seen my aquariums, and I just, in conversations with him, talked about things that interested me. And he heard that Forrest Rower was going to be giving a lecture at UM, and he invited me to see it. And I had never heard of him. And when I was at the lecture, I was blown away because it was a topic that I knew a little bit about. I had heard about or read about phages, which are viruses. And even, because it interested me back at that time in 2004, when Charles Delbeck and I were writing the Reef Aquarium Volume 3, I included a bit about that because I thought, gee, this might be interesting for controlling bacterial disease in our aquariums. And so there it was in a little box of thought. And that's now more than 20 years ago. So anyway, almost 10 years ago, I saw Forrest Rower give this lecture, and I thought, God, the reef aquarium hobby really needs to be in touch with him because there's some contradiction, and wherever in science you see contradiction, that's ripe for discovery. Because he got up there and he was telling the audience that, when reefs are microbialized, the algae grows out of control and you have this phase shift, and whenever you have algae in your corals, the corals die, and it's just bad. Algae and corals, bad. When you have these situation with excess dissolved organic carbon, the corals just do terrible. And I was thinking, gee, I could get up at the end of this talk and tell him, did you know that people use algae to filter their reef aquariums and the corals thrive? Did you know that people dose dissolved organic carbon and it makes their aquariums better? And I just thought, if I do that, everyone's going to not understand what I'm doing. I'm going to look like that asshole. And I decided I'm not going to be that asshole. And I kept it, it was on my list of things to do. And, you know, 1 year becomes 2, 5, 10. And all of a sudden, you know, I realized, oh, gee, you know, maybe he could come and lecture here. And so a few months back, I sent Forrest an email and I explained to him what I just talked to you about, that, oh boy, I work on long time scales. It's been 10 years I've been thinking of talking to you. Anyway, let's see, where is that? Yeah, I talked about the value of connecting with you in a lecture I gave in 2017, and you can see this lecture on YouTube. It's a bulk reef supply. They videotaped me in my little stories. I think that was at MACNA back in 2017. And I talked about the contradiction and, you know, what does that mean? Why is it that this observation that Forrest was making didn't seem to fit with our observation in aquariums? So I've had a lengthy back and forth conversation with Forrest by email, not by phone. And he's enlightened me on things that they've observed, and he's not surprised at the contradiction. He probably would have been humble and understood where I was going. I don't know if the audience would have if I stood up and said those things back then. So let's talk about definitions. You know, when you say the word microbialization, we are talking about microbes. Well, what are microbes? They're little bitty organisms. They may be single cells or colonies. Most of you also know that you have this tree of life where you have bacteria, archaea, eukarya, and so we're not just talking about bacteria. There's a lot of different critters. Critters not the right words, but these little creatures. And you can read more about this. I'm not going to dwell on it, but you'll notice that there's something missing in this picture. And it's kind of funny, you know, my background is in biology. And I remember from the beginning, from high school and in college, you know, there's sort of this segregation of microbes and viruses. We just don't know where to talk about them in the same subject. But the truth is, bacteria and other microbes and viruses are kind of like corals and zooxanthellae. They're not necessarily symbiotic, but they do live together. And there are things that happen in nature, in us, in our aquariums, in ecosystems because of this interaction. And that's what my lecture is about. And then we can talk about how Forrest Rower has elaborated on this since the discovery of phages, which is a little over 100 years ago. That's Forrest Rower. I have not seen him recently, but that's about what he looked like about 10 years ago when I met him or saw him at that lecture. And I should say that it's not just Forrest Rower doing this work. He has a lab at University San Diego, or excuse me, San Diego State University. And there's a whole group of people, collaborators and grad students that work with him. And this is an important paper that they published, and they've done many, many papers on this subject. I have that up there long enough for you to photograph it, and you'll probably want to look at that one. Also, he, as well with a co-author, has written some really interesting books on the subject. You may want to look into that as well. And here was a response, a boil it down in a nutshell response from Forrest recently to me in conversation by email. The main takeaway from our work is that the amount of labile organic carbon to oxygen ratio determines whether the ecosystem is microbialized, which in that situation is lots of organic carbon, lower oxygen level, versus viralized, low organic carbon, and plentiful oxygen. That's a key difference between states, conditions. And that would be true in nature as well as in our aquariums. Microbialized is bad for corals. Viralized is good for corals. The viralized system is characterized by lots of viruses, and we're talking about phages here, that kill the bacteria. In a microbialized system, the viruses are actually inside the cells as proviruses. And I'll show you a diagram where you'll be able to visually understand this as opposed to the words that may be making you sleep right now. That condition is called, I don't know whether it's lysogeny or lysogeny, but probably lysogeny. Pathogens are lysogens. Phages are beautiful. We tend to think of viruses as bad things, but these phages are really powerful. They are extremely abundant all over the place. Seawater contains approximately 10 million viruses per milliliter. There's a quote on one of Forrest Rower's websites where he talked about if you could take all of the phages and line them up head to tail, it's unbelievable what distance that would span. And I don't remember, I didn't write it down, but they are the most abundant biological entities in the sea, and we hardly talk about them. Most are bacteriophages that infect and kill marine bacteria. Viral abundance is tightly linked to the host bacteria, and viruses are most abundant not just in bacteria, but in phytoplankton and cyanobacteria. Viruses recycle nutrients together with organic matter in a process called the viral shunt, and if you're interested in that, look up viral shunt. It's not enough time to go into it here. So how do you even know they're there? Well, there are ways of measuring for them. And then studying viral community dynamics. They use processes called viral metagenomics, virus to microbe ratios they're measuring. There's biochemical diversity via this metabolomics, and I think one of the speakers yesterday mentioned that. Microbial community dynamics via metagenomics. Okay, so there are ways of looking for these things by sampling and testing the water. Why does this matter? Or why does it matter to me? Why should it matter to you? Well, development of husbandry techniques, a recipe to avoid microbialization, provides a way to avoid coral disease and death. And why do we want to avoid coral disease and death? How many of you have set up a stable, wonderful reef aquarium, and then suddenly one day, and you don't have them in your tank, one day, poof, a coral dies, and you don't know why. It happens. Now, I'm not saying that getting microbialization under control is guaranteed to stop that from ever happening again, because there are critters, things like them, that can come in there. There's always something, right? But I think it's critical for us to understand the processes of microbialization, and if we can truly control it in our aquariums, then that's just one more tool in our toolbox and husbandry technique that will promote success. And bottom line there is that this is a key to the success of coral restoration and coral farms and coral banks. You know, we can put billions of dollars into trying to preserve genetic biodiversity of corals upland and coral farms, but if we're missing that key husbandry technique, we're going to be successful 99% of the time, but boom, you might miss you know, something that promotes microbialization. So phage therapy is a real possibility as an alternative to the use of antibiotics and synergistic co-treatments for controlling bacterial disease infections in corals. Phages can be cultivated with probiotic bacteria and dosed, administered when you have a disease event. So that's a practical application of understanding what Forrest Rower was talking about. So if you're still awake, are you beginning to understand that this is ultimately kind of a black box problem. I've only just sort of scratched the surface. It really can be much more complex. Getting back to the subject of DOC, dissolved organic matter comes in many forms. We know that because we utilize different means of controlling dissolved organic carbon in our aquariums, be it activated carbon, protein skimming, and other forms of chemical filtration. Dissolved organic carbon released by ungrazed fleshy algae supports copiotrophic, potentially pathogenic bacterial communities, ultimately harming corals and maintaining that state of competitive advantage for the algae. How many people have ever seen this in a reef aquarium? The algae bloom, and no matter what we do, we can't effing control them. Julian told you you can just drop the phosphate level down. Algae still grows. Guy at the pet store says, oh, control your nitrate level. Algae laughs at you. There are these phase shifts that happen in our aquariums, and they are not just nutrient controlled. Truth is, the nutrients are important, but they're not the whole picture. There is a community ecosystem thing that can shift back and forth, and it's related to this topic of microbialization. Reef algal cover is positively correlated with lower concentrations of DOC and higher concentrations of microbes. Okay, you got that. That's a key thing. Algae blooms, releases DOC. After that DOC is released, microbes bloom. And it's the microbes that are supporting this balance. The viruses are there, but sometimes the viruses go into the bacteria and stay there. They don't kill the bacteria. And usually when that happens, the bacteria become virulent and kill your corals. Algae then grow over your dead corals, and it stays that way. Same thing happens in nature. Microbialization refers to the observed shift in ecosystem trophic structure towards higher microbial biomass and energy use. On coral reefs, the proximal causes of microbialization are overfishing and eutrophication, both of which facilitate enhanced growth of fleshy algae and give a competitive advantage to that fleshy algae over the calcifying corals and coralline algae. I wrote a book for aquarium keepers about how to control algae. Many of you are familiar with that, and it's a topic that every reef aquarium hobbyist is keenly aware of. I wonder if you've thought about this now that I've been talking about microbialization. We tend to think of herbivores as algae control. Think about this. Herbivores are microbial influencers. Wow. Think about diadema in the Caribbean. What does it do to avoid microbialization? Its grazing is key. It's not just extraction of the algae. It really changes the whole dynamics and makes the phage have a competitive advantage. And you can do that in your aquariums too. This is a quote from Forrest in an email conversation. We're particularly interested in how DOC and oxygen levels influence the balance between lytic and lysogenic viral cycles. If you're not familiar with that situation, it's a fairly complex drawing, but let's see, will this work? Do I know? Is this the pointer? No. Okay, I don't want to make, how does that work? I don't know how to get the pointer to work, but in any case, you see the two circles, the blue circle and the red circle, so that's defining the difference between lytic and lysogenic cycles. The lytic cycle, that's what you're looking for. That's where the phages are blooming and destroying the bacteria. The lysogenic cycle, that's the microbialized state. That's where the phages just sort of end up in the bacteria and make the bacteria virulent and multiply with the bacteria, tell the bacteria, here's what you're going to do now. And it often is a result killing the corals. So we want to get things toward the lytic cycle. I guess I've explained that. So a bacteriophage in lysogeny integrates its genome into the host bacteria's DNA, and thus it becomes what's called a prophage, and replicates passively within the bacteria. So as the bacteria are replicating, the virus goes with it. It enhances the pathogenicity, making common bacteria virulent. I just explained that. So why it does that, I don't know, but that's just the way it works. And as a side note, aside from making it more pathogenic, it enhances the survival of the bacteria. So as I mentioned earlier in the talk, you know, this virus and bacteria relationship in some ways is symbiotic. In the lytic cycle, the virus attaches to a receptor on the host bacteria, injects its genetic material, and then replicates using the bacteria's protein and nucleic acid, and then it just destroys, you know, it uses that stuff to assemble new viruses and destroys the bacteria cell, bursts out, and then keeps replicating, going and finding more bacteria. Alternate states. It's kind of interesting how algae themselves have alternate states of reproduction, and then you have this lytic versus lysogenic cycle with the bacteria and the phages. How many of you before coming here today were aware of this? Yeah, quite a few. That's very good. Okay, another quote from Forrest. The idea, in other words, what you want to do or what we'd like to see is to encourage virus-driven top-down control of the microbes and microbial communities rather than allowing long-term integration and dormancy. And the integration and dormancy is the lysogeny. Toward that goal, here's where you as Aquarius ears are perking up. One of the simplest and most effective tools we've used is elevated oxygen. Okay, well, hobbyists know it's important to have good gas exchange with our aquariums. And we know that we try to maintain saturation of oxygen. Well, maybe what we need to be doing is going even further. Running a commercial oxygen concentrator alone can shift the system, increasing viral phage activity while suppressing heterotrophic bacteria. Wow. Okay. First thing I thought of when Forrest mentioned that was, geez, back in 1988, when I was working for John Burleson, he built an oxygen reactor, which he got from a design in a book by Lemkemeyer that he got from Mike Paletta. I still remembered that. So I wrote to Mike Paletta after I got this email from Forrest, and I said, can you dig up your old Lemkemeyer book? And he said, oh yeah, I'll get it. Hasn't found it yet, but we'll get it. I want to be able to talk about it. Now, what's important to know is that the Lemkemeyer design had an additional feature. It was pressurized, really, really interesting. So there's room now for new technology, right? Oh, we love technology. In any case, the use of these oxygen concentrators, which are made for people who are, you know, oxygen challenged, you can buy this on Amazon. That alone could be used to help deal with microbialization. Of course, I went and bought one. I don't know why it hasn't been delivered. That was a few weeks ago. Maybe tariffs. In any case, I do plan on doing some initial testing to see what I think. And some of you probably, if I don't mention it, will then ask me, well, what concentration of oxygen are we looking for to achieve this? And I don't know the answer to that just yet. But here is what I wrote to Forrest. Hey, are you simply bubbling it in the tank using a protein skimmer for contact? Do you have any control equipment to turn it on and off? And I thought you'd be amused at his response. He just said, we just bubble it directly in the tank, adjust the flow rate to get our target oxygen concentration. Don't know that target level for the experiment. And the controls, well, serially deployed undergraduate volunteers. SDUV. Nicole, do you know what SDUV? You know, undergraduate volunteers in an academic situation. I thought it was very funny. Something else, just a side thing, there are multiple websites that Forrest and his team have worked on, and one of them, look up, is this phage to shark. And the emphasis there is that, you know, we all have heard that overfishing tends to promote microbial or promote the phase shift to an algal reef. And maybe, you know, unless we really study the energy shifts in ecology, we don't necessarily understand that. But this website really makes that clear, how sharks are biological sinks. You know, you have all that energy go from the you know, first consumers all the way up the food chain, and the end of the food chain, the apex predators are absorbing all of that energy. Well, interestingly, you know, we tend to think of that food pyramid and apex predators as being big, and they are. But what we just talked about, phages, are the microscopic apex predators. So this whole process goes to that side and that side. That's another blow your mind moment. And well, I've just explained that. Look at Phage to Shark website, and it's interesting. So as I've mentioned, microbialized ecosystems, lots of organic carbon, low oxygen. The phages are inside the bacterial cells, and that's bad for corals. Viralized, low organic carbon, plentiful oxygen, and that's good for corals. And the phages are killing the bacteria. So we already have husbandry techniques that influence this to some extent. I mentioned algae filtration, and you might think that Forrest Rower's team would be opposed to the use of algae filtration. No, in fact, when I brought it up to him, he said, we use Chetomorpha. So, and we find it to be very beneficial. So, you know, not all DOC are bad, and algae in the presence of coral is not a death sentence. You need to understand that. Carbon dosing, they haven't experimented with that. I think you know, a bottom line is, and some of us have found it, is that carbon dosing, while provably beneficial, does have some risk. Biopellet filtration is something like carbon dosing in a solid form, promoting bacteria to assimilate nutrients. There may be some risk in that the same way. Protein skimming is a way of dealing with some of the dissolved organic carbon. It also removes bacteria and phytoplankton along with dissolved organic carbon. So it's having some influence on the microbial condition in our systems, you know, aside from affecting nutrient levels as well. Mechanical filtration, nowadays it's quite popular to use not only socks, but also these rolls. That has some influence on these conditions. Chemical filtration, whether it's activated carbon or otherwise. UV sterilization, what does that do to phages? I don't actually know the answer to that question. It certainly does have an effect on bacteria levels in the water as well as other microbes. Ozonation, likewise, affects DOC as well as microbes. Peroxide dosing affects microbes as well as algae. And chlorine dosing, which I bring up because somebody here, Bob Stark, many of you are probably aware, talked about that. And I remember when I was reading it, I thought, aha, that's a really interesting idea. And I wonder what that does to microbes in the water column. And yeah, really, you know, you might think, well, why would you do that? There's a lot of influence that could have, done right, correctly, small quantities, could push systems into the right direction. Here's a thought. A lot of us add phytoplankton to our aquariums. Could phytoplankton feeding actually be virus dosing? Do we know whether the phytoplankton cultures that are available have phages or not? Has anyone looked at that? And if they do, that's interesting. If they don't, should they? Okay. What about the ugly phase that we all experience when setting up a reef aquarium? Is that a microbialized phase? When it stabilizes, is that the phase shift? Could we push it by adding phages? Microbialization in aquariums. So we've been focusing on the effects on corals. What about fish? Diseases in fish. Could these shifts be affecting them? I don't know, but maybe. It certainly seems possible. And that's an area for study. I don't bring it up to just throw out a red herring. I'm just bringing it up to say, if you're interested in this topic, this would be really interesting to look at. You know, the presence of cryptocarium, amylidinium, uranema could be tied to DOC viruses and all of this whole process. There could be some connection. What about the spring effect? There's Sanjay saying, this is how I know spring is here, the annual Spring RTN special. Remember that, Sanjay? Not so long ago. Yeah. Don't you just wish it was just that? Okay. So there's actually a paper talking about this. Hobbyists have been talking about it for years. It's not just a full moon and everybody's driving like crazy effect. There's really something to it. This was a paper, I don't have the link there, but I'll keep it up long enough for anybody interesting. You can look that up. And I want to just, I'm sorry I don't have the reference or link, but I'm going to show you some of the quotes from this paper that would be of interest to you. Could it be an allergy, like hay fever? The answer is no, it's not. Corals don't have an adaptive immune system. But that doesn't mean that the effect of the pollen isn't real. So let's just look at some possible connections. And this is a quote from the paper. Recent evidence suggests an aquatic origins to viruses of the order Articuloviralis, which includes influenza. And that occurs in Cnidaria, including corals. Herpes-like virus has been known a long time, are very common in corals. A bacterium, Serratia maricens, is known to cause issues such as septicemia, endocarditis, and osteomyelitis in humans, and it causes whitepox disease in Acropora palmata. And even plant pathogens are found in the marine environment. So the same one that causes tar spot in sycamores, it's a fungus, has been shown to share the marine biome and cause similar disease known as dark spot syndrome in corals. So, you know, could pollen carry something that when it lands in your aquarium is going to cause a problem? Yes. Could it also just lead to microbialization, a shift? Yes, possibly. So there's more work that needs to be done. This is from that paper, and they just show the potential pathways. They even talk about an increase in reactive oxygen species that could be promoted by the pollen abundance, you know, in the water. So it's still a study to be teased out and worked out what's going on there, but it's related to this topic. Saharan dust, it's been known for quite a long time that there's this dust that blows from Africa across the Caribbean, settles, supplies micronutrients as well as microorganisms, promotes blooms of sargassum, which then flow into our coasts all along the Caribbean, including Florida. Probably promotes blooms of red tide in the Gulf of Mexico. So anyway, it's not unreasonable to hypothesize that a similar process isn't occurring with our home reef aquariums from pollen. What can we do with all this information? Well, Joe, I heard that you were working on some kind of a device.
I told you not to say anything.
I missed that.
Julian.
Yeah. So anyway, there are things that we could be looking at. I mentioned the oxygen concentrator as something that could be utilized. Do we need to be using an apogee oxygen meter? Well, I don't know. We haven't been using it for decades, but maybe it's something that we can utilize if we want to bump up the oxygen above saturation, whether that's permanently or temporarily. And it's a relatively inexpensive meter that you could use. And by the way, what happens to the oxygen concentration in the air in our homes and closed buildings diurnally, you know, during the day and night versus seasonally? It's an interesting question, but the answer is probably not much because, you know, big changes in oxygen concentration, you're going to feel them. You're going to get tired. You're going to pass out. So, but that doesn't mean that there's no effect. And I don't think anybody has studied this. And so there could be rare exceptions where there is a measurable difference. And the amount of oxygen that dissolves in seawater is really only a very tiny amount. So it doesn't take a big change in the air to have maybe a bigger change in our aquariums. And somebody should perhaps look at that and say, nah, there's nothing to this. Or yeah, maybe there is. You know, Craig talked about the effect of CO2 accumulation when people were looking at Greg Scheimer's aquarium, and that's a real phenomena with very big effect. But it seems that oxygen concentration in closed spaces doesn't vary a whole lot. Anyway, this is talking about the solubility of oxygen in seawater. Of course, oxygen solubility in fresh water is higher. If I had the pointer, I could show you what column to look at. You want to look at the milligram per liter column under the first section there, and go down to, say, 25 degrees, and you see 6.6 milligrams per liter. And that's about where we're at saturation in our reef aquariums, which is not a lot of oxygen. So when the power goes out, it doesn't take long to go from 6.6 to a lot lower. However, further to the right in this diagram, you're seeing look at the milligrams of mercury, 760 to 1520. If you double the pressure and then go to 25, you go from 6.6 to 13.2 milligrams per liter. So increasing pressure does put more oxygen into the water. So Lemkemeyer's oxygen reactor was doing that. And John Burleson built one of those things. And it definitely, I remember he had the most absolutely amazing, spotlessly clean and corals happy reef aquariums back at that time. And so it interested me that this is something that perhaps that was part of what he was doing. I mentioned Craig, and yesterday he gave a talk on and brought up his diagram, and since he created a new one, I thought I would show the old one. But not to piss him off or anything, I had put this in the talk before yesterday. I didn't know that there was going to be a new one. I took a picture of it yesterday and I could have slid it in, but I decided not to. I thought I'd leave it here. It was still pretty damn good when we put that in the Reef Aquarium Volume 3. And the reason I bring it up is to point out that that box is referring to a zone. You stay in that zone of calcium and alkalinity and your corals are going to grow like Joe's corals. Unbelievable. Ooh, stay in there. And what is that zone? What do we call this thing when you have the intersection of two different parameters and they relate to each other? I don't know. And are there other types of parameters that create a zone, a box like that, where if we stay there, the conditions are going to be right? And I can tell you the answer is yes, there are. And that's part of aquarium husbandry and part of understanding how to make your tank look like Joe Waiulo's, right? Even if he has them in there. So one of them would be labile organic carbon and oxygen ratio. That's a zone. And we really didn't think about that until this lecture. Okay? And now we also know that temperature is related to that, so it's really a 3-parameter zone. Nitrate to phosphate ratio. Now, that doesn't necessarily directly tie to microbialization, but it is a zone that many of you know about. And I recommend looking at this lecture that my co-author, Charles Delbick, gave. You can find this on YouTube, on the High Tide Aquatics YouTube channel. And he's talking about the nitrate to phosphate ratio and what he discovered over time observing corals in the Steinhardt Aquarium Reef. Very interesting. My comments to him when he was looking into it was that, gee, if this is a real thing, that will be great because we can really then have a husbandry technique and develop a recipe that will make us more successful. So, very interesting topic. And Charles also, because he's a really good guy, heard that I was giving this lecture. And when I was, I think I was on my way to the airport to come here, he sent me the link to this paper, which was just published this week, last week. Cell Reports and Sustainability Journal. It's not a paper where research or testing was done. It's really more theoretical discussion. But reef water microorganisms as diagnostic indicators for coral reef ecosystem management and sustainability. Eli, where are you? Yeah, this is like right up your alley, right? Did you sponsor this? Yeah, anyway, really, really interesting paper. That gave me the ability to put in some more graphics because my whole lecture has been just words on there. Sorry, guys. But looking at methods of measuring and studying, monitoring microbes in the water, whether this is, in this case, on coral reefs, or hey, we can stretch and think about our aquariums, right? And why it's important, how to do it. Probably too much to talk about there, but take a look at this paper. It's available free, no paywall. But yeah, look at this, types of microbial indicators. They talk there about Prochlorococcus, which is an abundant microbe in the sea. When it's abundant, that's an indication of healthy oligotrophic water. When it's rare, that's indication of pollution, disease, other abnormalities. Heterotrophic microbes in high abundance, that's an indication of pollution and disease. When they're low in abundance, it's a healthy condition. Synechococcus, let's see, I guess that's a variable one. And photosynthetic heterotrophic microbes, boy, it looks like it goes both ways with those. You'll have to read the paper to understand what that really means. Okay, and so this was another idea talking about decisions on what to do and monitoring and decisions that are made for policy for reef protection using measurements made in the water to help aid those government decisions. That's something that academics tend to talk about a lot. And then this paper was adding this to the toolbox. Usually you have researchers who do, lay out quadrants and measure how many corals there are, and, they come up to develop policy of what you need to do. Here they're just saying, oh, no, test the water, and then we can have answers, right? Okay, and developing a microbial health index. Clearly, Eli, again, right up your alley. Okay, I won't dwell on that more. Here was just another thing that I thought I'd mention, and it may have some tangential connection to microbialization. There is a very rare fish that is a hybrid Scopus and purple tang that I put into a reef system where obviously conditions were not great for it. It was a place where I knew that it wouldn't jump out, which I was glad, but clearly it started to develop head and lateral line disease. I see all the time people post pictures of zebrasoma with the fin erosion like that, and they're like, gee, I don't know, there must be some aggression. I never see anybody attacking or biting them, but the fins are going away. In case you don't know, I think most people know here, that is not aggression physically. There's no attack going on. That is a symptom of head and lateral line disease. And unfortunately, the lights are on too bright here, but if you look where the lateral line is, right in the upper middle, you see there's a bit of a white line there? It's already starting to erode, okay? Now, believe it or not, Reef Notes was not my first foray in writing in Freshwater and Marine Aquarium magazine. The first publication in Freshwater Marine Aquarium magazine was when I was 15 years old. Don Dewey, the editor, picked up an article that I had written for the Florida Marine Aquarium Society where I talked about curing this. So it had head and lateral line disease has been a mystery and a bane, a problem for hobbyists for a long, long time. And I was able to cure it in a hepatis tang that I had when I was 15 years old, and I thought, gee, people should know about this. And so I wanted to demonstrate how, I can still do that and how quickly I can do that. So this was May 14th. And here's the same fish just a few weeks later, June 5th. I took him out of the tank where he was, put him in a different tank where I knew it would heal. And I was even surprised at how fast it went. And I did it with this fish, so you know I'm not lying, because there's only a few of these fish in existence. They're about $5,000 each. And trust me, that's the same fish. Now, I'm not going to stand up here and tell you the one single thing that caused that head and lateral line disease, because I know very, very well that there is not one single thing that causes HLLE. There are many things that can lead to those symptoms, okay? And if you ask me what was it about what I did that cured it, I have to confess that from a scientific point of view, I don't know. What I do know is I can reproduce this. If you want to send me a fish, I can cure it, okay? And I do know that the papers that talk about dietary changes improving it are correct. That sometimes that's it. And not just what you're feeding, but oftentimes how much, the amount of nutrition. But there are also papers that talk about water quality conditions that affect it, and those are absolutely correct. There are water parameters that you can create that will make that happen and reverse it. Why? I don't know, but it's true. There is also psychological stress effect. You can have multiple zebrasoma in a system, and the odd man out, the one that's being badgered and bullied, will develop HLLE, and the other fish won't. And that thing is a kind of a stress plus dietary, because even though they're eating the same food, because the fish is bullied, it's not eating as much as the bully. So it's a really, really complex subject. And it may also have to do with microbialization, because one of the other factors that's been discussed is the use of activated carbon. So there is some effect of DOC on the start of HLLE. Someday we'll know a little bit more about it. I'm almost done, I promise. This is the last slide, and I'm glad that Joe mentioned Terry, and I wanted to, right at the end, since I'm the last speaker, bring up Terry. This is a photo taken at Greg Scheimer's aquarium, and both Terry and Greg would have loved this event. Yeah, and so in any case, you know, and that goes back to a long time ago, and you know, what a great moment for us all. And it's a pity that Terry couldn't have been here, because he really, really would have loved it. Can you imagine Bob Stark and Randy Donovitz so young? Right? Yeah. Anyway, so I was glad for all the things that you had said, Joe, as well. There's not much more I could add. Terry wouldn't want us to blather on about it, but he definitely was a big part of making the connections between the hobby and the academic world, and especially making sure that new information was disseminated as quickly as possible, and keeping things true and honest. And so it was a pleasure knowing him as a friend, as a hobbyist, and working with him in Aquarium Frontiers to that end, doing that. So that's the end of my talk, and you're welcome to, you know, ask any questions.
Thank you, Julian.
Yeah, thanks.
So I'm going to double down and say, if you want to reach Julian, 218-206-1225.
Kick your ass.
So, okay, before we get to questions, I'm just going to put this up here front, some schedule stuff. Okay, we're going to take a break after Julian's questions, and then we'll have our final panel of the day.
Yeah.
Really important that everyone who is going to be on the cruise needs to be at the Campus Recreation Center by 5:10. You must be there at 5:10. So we're going to take Julian's questions. We're going to have, if you're a reef keeper, after Julian's questions, please come up front. That was, I know you're really clueless, so yeah, that guy. Yeah, you talk to your partner there. So, okay, so yes, so don't run out of here after Julian's questions. But once we are done, 5-minute break, panel discussion. If you need to boogie out to get to your room before the evening's events. And of course, everything is here in your scheduled packet. So I'm going to take just one more minute before we get to Julian's questions. If you are someone who helped organize this event with the Roger Williams and all the phone calls, would you please stand up for a minute? Oh, there's more of you out there. Just stand up for just 1 minute. Just so people know that this was a team effort of a lot of people. All right, fair. This has been a fantastic event. So, okay, Julian, so the questions for Julian. Okay, I'm going to run around. We'll do maybe 3. I see 3 hands. Those are the 3. All right.
I have 4. Hey, Julian, you were talking about Forrest saying raising the O2 levels.
Yes.
Do you remember offhand what he was aiming for?
That I said that I know that's going to be the first question you ask, and I don't know.
Okay, and I shouldn't have.
Yeah, I don't.
You part the question.
That's okay.
Damn
I don't know if the
Russ.
mic is working.
Damn it.
Yeah.
Oh no. So Julian.
Yes.
As one of the high altitude reefers here,
Yeah.
I have lower pressure of oxygen that I'm dealing with.
Yep.
That means my headroom for temperature is lower and salinity, all of that affects. Right? Are you recommending that I break out my 30-year-old Sanders ozonizer?
Well, ozone dosing is not the same thing.
I agree, that's what
Yeah,
I'm curious.
it's not. Damn it, I don't know if the microphone is working here. Is that working? Yeah. Oh, now it's working. I don't understand. Anyway, yeah, and I did bring that up with Forrest. I was hoping to elicit an elaborate response from him, and he kind of went, yeah, it's a black box, isn't it? Meaning that someone needs to really look at how different it is. Ozone, of course, has antimicrobial effects. Boomer wants to say something about it because he knows. Can we get a microphone to Boomer? Or talk loud, Boomer. What absorption?
Pressure swings absorption. They've been around for about 50 years. They're not new. They were invented by a Japanese guy a long time ago. You have 3 canister filters that are filled with zeolite. You pump in air, each canister filter takes out certain amounts of dissolved gases. The pressure swing and kicks that stored gas out of the zeolite, and they're all ready to go again. You see people walking around hanging from the side, that's what they are.
Yes. That's an oxygen concentrator, is what he's talking about.
It's taking all the gases out of the air.
Yes.
Nothing left but oxygen.
Yes.
I was selling them to Tony Wagner.
Yeah.
I had a concept. We had one in 25 dollars back in the early 90s.
Yes.
It was too expensive back then.
Yeah, I think Mitch Carl told you to do that, didn't he? Yes. Boomer doesn't get the joke. I'm sorry. It just so you understand, everything, latest cutting edge, Mitch Carl did in the 1990s. So you have to follow along with that. And Tony Wagner. Okay. Well, I didn't know that you were doing that, and if everybody had known about microbialization, then you might have been successful with that. There would have been a demand, right? Yeah. Anyway, there's something that needs to be studied and to see. I think what would help, first of all, would be for Forrest Grower to come and lecture at a place like this, and also to publish what he's doing in aquariums with illustrations and showing. All I have is the conversation. I haven't seen what's being done there in his lab. I may need to just go visit in my spare time, right? But in any case, I thought you were going to answer the question of the difference between oxygen versus ozonation. Yeah, anyway.
Show down here. Hey, Julian?
Yes.
Yeah.
Right. Joe.
Yes. So I have had the pleasure of being the lead on seahorses for the last 2.5 years.
Yeah.
And a while back, I heard somebody using oxygen concentrators to bump up the oxygen levels in the system.
Yeah.
To help alleviate all the lovely things that we have to deal with seahorses.
Yeah.
So I started about 2 months ago. I've been running the system at 140% coming out of the skimmer.
Yeah.
Exhibits running, habitat. I'm trying to keep up with it. Habitat is running at about 124%, and the returns at about 118%. Too soon to tell anything, but that's the numbers we were shooting for. I know you didn't have a number, but gut meter.
Yeah.
If you were dealing with lovely seahorses like I am now, and trying to alleviate all the situations that they have, what number would your gut meter shoot?
I don't have the experience to be able to tell you. Obviously above saturation somewhere, but I don't know. And I don't know whether there is a certain, obviously there must be a certain percentage where it's too high. So I don't even want to begin to guess because I don't know.
Okay.
Yeah.
Fair enough.
Quick question.
Well, quick thing to ponder, not that anyone wants to hear anything more about Joe's tank.
Yeah.
As you contemplate the massive growth in there and all this viral stuff, you're the only person I know that uses natural seawater that's not sterilized.
Right.
Lots of us use in that seawater, but we filter it and sterilize it, and might be something to ponder
Yeah.
about the effect of
The virus, the phages that are in that water.
chlorine,
Yeah. Yeah, you don't chlorinate it? No. So you've got phages.
It is ozonated.
Oh, well, the
But
ozonation.
very, very low dose.
Okay.
24/7 on the tank itself.
Okay. Yeah.
All right,
And
Eli
calculus.
is getting the last question.
So I wanted to share the observation first that, about your pollen story.
Yeah.
One of the signature terrestrial plants in our eDNA testing of aquaria, and I can't wait to go home and look at seasonal patterns of this, as well as correlations with coral health.
Yeah.
So stay tuned, we may have a data set on that already.
Great.
And my question for you is, you discussed that microbialization can impact the microbial community structure. I'm curious if you found any resources that describe those characteristic changes in the structure that we might use to interpret microbiome data.
I haven't taken the time to look. It could be that somebody in the Rower Wegley lab has, and that would be the right team to contact to ask. Yeah, all I am today is the messenger on this topic. You know, I've been thinking about it a long time, and I tried to condense it into a way that was, you know, understandable to, you know, hobby level, even though I know everybody here is pretty much way above hobby level. Right? Yeah. Yep.
Julian,
Okay.
do you know what level of O2 Morris is going for?
That's the second time we're asking that question, even after I said you were going to ask. I love it.
All right, so it is 4:03. Please be back in here at